Hello, hello. Good morning, everyone. It's my distinct pleasure to introduce our speaker today, Dr. Shujun Ou from the Department of Molecular Genetics at The Ohio State University. Dr. Ou earned his PhD from Michigan State University and completed his postdoctoral training at Iowa State University and Johns Hopkins University. His research sits at the intersection of plant genomics and biomechanics. His lab focuses on the evolutionary dynamics of transportable elements, or jumped genes. He investigates how these elements drive genome evolution and the critical role they play in helping plants adapt to environmental stress. For those of us working on genomics, you are likely already familiar with his contributions to the community. Dr. O has developed several gold standard bioinformatic tools, including EDTA and LTR retriever, which have become some essential resources for genome annotation. We are very excited to hear about his latest work today. Please join me giving a warm welcome to Dr. Shujun Ou.
Thank you so much for the very nice and warm introduction, and thank you for inviting me, the students inviting me to come here. And I'm glad I can make the trip today. And this is a summary slide for my research program at Ohio State. So the lab is entirely focused on transposable elements, and we have three major focus. First is annotation. We need to know where they are, because many new species we do not have the annotation that's corrected. And then the second is when and how the TEs contribute to gene regulation and gene regulatory information, because TEs they use RNA-2 to transcribe and then the response to regulatory cues. And when does that information become the regulatory component for genes is of interest. And since TEs insert into genomes frequently and get deleted also frequently, how do those events become faithful recorders for genomic evolution, including speciation, regression, and divergence? So we developed many tools, including several that for annotating TEs and also analyzing TEs. And we're also adapting new methods like FibroSeq to assess the competence openness of the status. And all of this development is focusing on how do we understand biology better, including regulation and evolution. But today, as I'm invited by the student body, I want to resurrect my PhD project from my last chapter, which is not about transposons. So I started this project even before my PhD, and I was from my undergrad project, and then I asked my PI, can I do something about PopGen in Rice? She said yes. And until now, this project has not been published. So the first lesson learned is finish your work before you leave. Otherwise, they won't be done. They will be get perished. So this is an effort I try to complete it, and hopefully I have some progress this year. And so you're eating rice today, but you may not know rice. There are two types. One is called japonica, which is sushi rice, short grain, or in Chinese, gandao. And the other type is long grain, indica, or in Chinese it's called xiandao. So you said even in Chinese there are two words describing the two rice, which means they have been exist for quite long. And you can see the phenotypic differences between japonica and indica. And Nippon bear is the reference rice genome, which is the typical japonica, and 9311 is the typical indica, and you can see the morphological differences. So since they are all rice of the same species, how do they come together and as like two very drastic different, we call it subspecies level? And the question came with the initial domestication. So that involved converting a grass, like what you see on top of here, and into a very highly neat plant and highly managed. So this transition, these key transitions, we call it the initial domestication or key domestication genes. One of them being phosphate growth 1, PROG1,
I hope everybody realizes that's hugely humorous.
Yes.
Right.
It's quite humorous, and and this is not about some male organ, but it's about the statue of the plant. So as a grass, you actually want to expand as much as you can to harness your space and light. But as a crop, you want to be tidy, compact, and then easy to manage. So this transition from this lay down into stand up, the gene underlying it is key. And there are several genes like that. And if you look at those genes, they seem to have a shared allele that's suggesting they are domesticated only once. Yeah, shared between indica and japonica. But if you look at other important genes, such as controlling when they flower, how tall is the plant, and whether they have reproductive isolation or the grain size, you start to identify there are different alleles. Like in indica, they're mostly ancestral allele, and in japonica, they're mostly derived. And that suggests they've been domesticated twice or more than twice. So how does the two contrasting evidence come together? And that has been a debate, whether one rice was domesticated once or twice or multiple times, and which was the initial domestication. And if you look at more than two genotype, you see these are two plates of rice in general. One is the indica plate, including Ausch, and then the other plate is japonica, suffering into tropical and temperate japonica, and even fragrant rice. So the whole genome level or multi-locus level also suggests the genome background came from two sources. And if you look at the genetic diversity in nucleotide, diversity in pi, disappear in indica and japonica, and in their more closely related white rice. They have reduced diversity and also increased population differentiation noted by FST. And you can see the indica and japonica has such a high FST that is even higher than some species. So that is like a very drastic and very different evolutionary background. And that how do they incorporate with some key domestication genes and some important genes, and how do they answer? So the research question I attempt to answer today is how many groups of rice we can identify, and what is the relationship between let me see if I can get this in here. Okay, great. And what's the relationship between major rice groups, and how many times was rice domesticated? What were the population dynamics of different groups? And when and where was the domestication of cultivated rice? So I was very interested in and still interested in answering these questions about rice. So the opportunity came around the 2010s. People started to sequence many rice. There are 3,000 rice genome sequenced or at 14x here, and then 446 wild rice sequenced at this paper at 2x coverage. So at that time, I decided to let's combine them and to have a full picture of rice domestication. So combining them is 17 terabytes and of raw data, and the cultivated rice came from different origin, including China, South Asia, Southeast Asia, Africa, America, Europe, and East Asia. And the wild rice has a GPS, mainly located in southern China, South Asia, and even in Australia. So, so combining this data allow us to look at both the wild direct ancestor and the cultivated rice and with different populations. So I used the GATK best practice back in 10 years ago. And I had over 25,000 files. I processed them, aligned them, trimmed them, and then used the joint genotyping algorithm to call SNPs and indels. And the joint genotyping is basically you align all the individuals together so that even those low coverage samples can benefit from other individuals and then give you more confident SNP calling. And of course, there are lots of false calling of SNPs. And then I have several filtering steps. One is called the variant quality score recalibration. So basically, this is a classifier. You use the NCBI SNP database that you know some are true, some are false, and then you train your classifier on your raw data and classify them into two categories. And also apply some hard filtering, including read number coverage, strand bias, mapping quality, etc., to identify as high quality as SNPs as I can, because noise will give me wrong answers. And then the first problem comes is the imbalanced missing data rate. Remember, the virus was sequence 14x and copy rice was 14x, and this is 2x. And you see a drastic difference of missing data rates. So that this is a problem. So I decided to do imputation. So basically, you use the haplotype of other variety that with coverage to infer the missing data of the focus genotype. And the imputation R square is the one that you use to evaluate the outcome. It's basically how the correlation between your missing data and the imputed data and the real data. So the higher the better. And for those low in the allele frequency, because you don't have a lot of observations in your population, and your inference tend to be less accurate. So after filtering, I just keep those with high confidence and retain those with high imputation R square. So to test whether the imputed and filtered SNP set is in high quality, I use other data sets to cross-validate our core sets. So I use the published data sets for copyright rice, and this is for SNP arrays, and this is the white rice, and this is the a different SNP caller developed by our faculty here, Eric Garrison, using Freebase. So they tend to be a pretty low pairwise discordance rate. So that gives me confidence on the quality of the data sets. So overall, I have many raw SNP filtered by BQSR, and some filtered by hard filter, and some filtered by low quality imputing scores, and retaining 17.7 million of high quality SNPs and all this analysis will be based on these data sets.
Could you go back a couple of slides to your math filtration? It looked like it was pretty good at about 0.2 math. You basically are doing really well.
Yeah. So here.
Well, yeah, up there at 0.2 math.
Yeah.
Well, farther, yeah.
Yeah.
So couldn't you just say, hey, I'm just gonna live with 0.2?
We just use the imputation R square because each SNP has the R score. So I just remove those with low R scores.
R squared of what?
R squares. The R square is
So you're going all the way to R squared is. Oh, at any math, you're taking R squared above like 0.95?
Yeah, 0.95.
0.95.
Yes,
Okay.
yes. And because these low quality SNPs do not give us confident conclusions, so I just remove them. And we still have 7 million SNPs that is more than sufficient.
Okay, but you lost 4 million.
No, these are
okay.
the index.
So how many did you lose in this process?
In this process is 6.7 million low quality imputed SNPs, and I determine is more value to keep the high quality ones.
But overall, you lose 16 plus 1.6 plus 6.7.
Yeah.
But you still have 17 millions.
Yeah.
So
Yeah.
your answer to your question is lost like 20 million SNPs, but that's okay. Like, because you got still 17.7.
I'm just kind of wondering what's the trade-off. So you lost something for sure. You gained a pure data set in your opinion.
Yeah.
And did you test alternative thresholds to see what you were losing and gaining?
The alternative threshold is the different R square, like 80%. I will get more, but I will also get noisier data. So I decided to like 17 million is sufficient.
But I think this will become important when you're talking about transposons, because the minor allele frequency of a transposon could be a lot lower, right?
Yeah. So we have a raised hand. Please unmute your mic.
Oh, Megan.
You can just, that's the username.
You can speak, or they can speak, or you can just lower hand. No.
Oh, yeah, just lower the hand right there.
Okay.
That's the, I think that's that computer.
Oh, okay. It's also USB. Yeah. So yeah, definitely you will lose some data, and especially for those minor allele frequencies, minor alleles, right? And I decided if you include noise, and you also include noise in your results, and I decided 17 million is pretty sufficient and more than enough for 3,000 genomes. So I use this as the final call sets. And because the coverage is low depth, and I have to be rigorous on the filter, otherwise we have lots of uncertainty. Although Eric's method of three base consider the uncertainty, but that's too heavy for compute. So the lesson learned is when combining data from different origins, the data heterogeneity will become a systematic bias. So you have to consider that and trying to remove the bias. And the raw SNP cost must be inspected, especially like if you pull it up on IGV, you will see how noisy the data are and then filter rigorously. So it's better to be cautious here than to be inclusive. So and I want to answer two questions first. How many groups of rice we can see, and what's the relationship between them? So I use the unsupervised clustering, using k-means clustering, and use two sets. One is the whole genome four-fold degenerate size. So these are the SNPs occurred on the third codon of the third codon, and then do not change the amino acid coding. That means they are more likely neutral than the others. And also use random SNPs, and they give me the same topology. So both share similar clustering, and also want to know, like, are there any more finite or more fine clustering of more number of groups? So I use a PCA, and to determine, use a scree plot to determine how many PCs can best explain the variation of rice. Suppose you find a turn point that give you okay. They give you the optimum PCs, right? But there's no turn point here, or there's like a turn point is too small. So that suggests the rice population history is very complicated, and there's no optimum population. So I select 14 because it includes more than 93% of variation in my data. And then I use this admixture to understand how the backbones of the rice genome share or not share two different populations. And this is a supervised clustering using K from 6 to 20, and 14 being this one. And each column here is one rice genome, and with the colors suggesting they share similar background, and those do not, or with a mixed background. So this population structure and PCA suggests a very complicated mixture or maybe even breeding histories. Then I asked, what are these 14 groups of rice? We know 14 groups of what are they, and how are they related to each other? So I make a phylogeny using the four-fold degenerate sites, and then using the minor allele frequency larger than 1%, end up with 150,000 SNPs times 3,000 samples. So this is a huge data set, and then I use RaxML to compute the maximum likelihood tree with this GTR gamma model. And I did 1,000 bootstraps to infer or assess the uncertainty of the topology. So this is how they're related, and we see some subgroups of indica, OSH, and then subgroups of white rice, and then some subgroups of japonica and aromatic. And because this rice was sampled in different countries and in different breeding status, we can even know what are the representing groups. Like, for example, this indica 3A are mostly traditional, like not improved, just from farmers' collection, and they collected from Cambodia, Laos, and Thailand. So this is mainly South Asia, Southeast Asia varieties. And for this one, mainly came from China, and it has both traditional, improved, and promising varieties, suggesting some of the breeding efforts are applied to here. And this one, like indica 1B, has heavy breeding and also potential improvement efforts applied to it, and it came from Philippines and China and India. And for the US, mostly our temperature japonica and tropical japonica group A, and with lots of improvements.
Sorry,
So some of the branches are really high.
Yeah.
And you haven't labeled them, so they're not part of your 14Ks.
Yeah, so these are the mixture groups that we do not have.
They're way the hell up there.
Where here?
For example, right, where's the highest one? So the highest one, I think, is. What's the highest one? For example, right below N2, right below
Yeah.
N2.
Yeah.
The high branch, very high, where the 97 is.
Oh, here?
So that's not a mixture.
Yeah, this is the bootstrap confidence value. So suggesting they have a very robust topology based on the data. And you see the numbers are slightly varying between these public two groups because some of the samples do not have all the metadata. So we just plot in what is available.
Okay.
Yeah.
so just a question for you, do you agree that there's two main groups, and does these two groups reflect the species rather than domesticated and wild?
I will follow that.
Oh,
So
okay.
next one. Yeah, so if you notice like the 97, we just point out, and also 89 and also 91, so that has rather high support rate, right, by the bootstrapping. And we also use the Orisa species that are not related as roots to orient our tree. So that suggests, you see, let me move this up. So you see this OR3A as the sister taxa of Japonica, and that has a very up in the tree. So just suggesting maybe there's like only one origin, or you can see this star with the OR1 as the closest sister group as OSH and also other indica. So maybe there are two domestication, but yeah, this tree looks nice, but it has unclear or maybe suggest multi-origins. So we want to explore further. So OR3A seems to be the most ancestral sister group of all favorite rice, but let's put a question mark here. Let's view the tree as the unrooted form. So this is OR3A, closest to Japonica, OR1 closest to Indica, and other wild rice here. So the wild rice samples are located at the Japonica side of the tree. Does that mean OR3A shares the most recent ancestor with initially domesticated rice? To test this hypothesis, I used simulation. Like it looks close and has many wild rice here. Does it mean this is the initial population domesticated? So let's do simulation. So I simulate a sequential bottleneck events with 7,500 generations. And the domestication or selection happened at 1,000 generations, and the second bottleneck happened at 1.5 thousand generations. And each of the selection only select 10% of the data, so resulting in a 10% bottlenecks. So I label this as D1 and the second one as a D2. And then I sample this population. This was using SLIM. I sample the population and then make a tree. And 63% of the tree look like this, and the remaining of the tree look like this. And both of them have the white rice in the middle, and the D1 and the D2 on closer to the white rice. So that means the D2 tends to cluster with the white rice, the wild population, and same as here. And the D1 as an initially subdivided seems to be further away. So this suggests what we see here. Maybe the initial domestication was in OR1 that suffered from the wild group based on the simulation. So that is confounding to the phylogeny tree that we just observed.
And is the rice like does the so is the generation one here and
I will follow on that too.
Okay.
Good question.
just if they mature at the same time, is it
Yeah.
synchronous or asynchronous?
Yes, I will follow that too. Same good question. Yes. So the second question I want to explore here is what were the population dynamics of different groups And I talk about bottlenecks. So when you select individuals from your wild population to cultivate it, you reduce your genetic diversity, resulting into a bottleneck. And we construct the population size history using oops, sorry, we have some doodling, using SMC++, and for each of the subpopulation. So you can see each of the population share a drop. So the y-axis here is the effective population size. They share a drop in their effective population size, but today, and they distribute in different generations. Does that mean they have multiple bottlenecks for each population? Your question is right. And the generation time is different. So white rice is perennial, means they can grow with the same roots every year in the flower. Cultivar rice is annual, but with temperature japonica, typical is one crop per year because they grow up north. You don't have a strong light to support multiple generations. But for indica or tropical japonica, there are 2 to 3 crops a year. That means you can have multiple generations per year. So I correct for these generation differences and recalibrate the population history. And then you have a more synchronized time around 10,500, 15,000 years ago, 100, right? 10,000, 15,000 years ago, right? And they overlap between indica and japonica, and also with OR3B. But you see OR3A still way ahead. This is like 30,000 years ago, and this was the last Gaussian maximum time. So does that mean OR3A was domesticated way ahead? I don't think so, but the other populations share the same time. Does that mean it was only one domestication? It's also unsure because this is only overlapping, right? Not causation.
What the hell happened 3,000 years or 2,000 years before present?
These things, yeah. So this is like further domestication and improvements, but the raise in population size likely from the algorithm itself.
No, the drop down
Here.
to 800
Yeah.
effective size.
Yes.
That's nothing. That's like 2 plots, 2 rice paddies.
Yeah.
Maybe
So
10 rice paddies.
Well, you cannot translate directly number of, this is just like reflective of the genetic variability. It's not, you can not easily translate.
But something made it crash. There was no glaciation in this part of the world.
So
Possibly.
there may be further improvement, or there may be the distribution of rice being populating the larger area.
No, can't be. This is pre-industrial, right? There won't be
I was not in, I was not there, so I don't know.
You understand how that.
Yeah.
They went down to
Yeah.
It could be just like
But it's not like the rice is migrating from continent to continent.
We'll talk about migration later. We'll have that. But here we just focus on the initial domestication bottleneck, and they seem to share the same time, but not suggesting they may share the same events, but not decisive, right? So the lesson learned here is phylogeny supports OR3A as the sister group of cultivar rice, but the forward simulation supports OR1 as the sister group of cultivar rice. And then the population history suggests OR3A as the initial sister group, while indica japonica had a shared domestication bottleneck at the same time. So it's getting more complicated. So how many times were rice domesticated? I want to answer that. So if you notice the admixture plots and look back at the K equals to 14, for the OR1, they're not pure. You have Ausch has a significant portion in the genome, and also indica 3A. And for OR3A, you have lots of portion from temperature japonica, suggesting heavy introgression coming from cultivar rice. And this is actually happening because cultivar rice is strictly self-feeding, but wild rice, they open pollinated, and they usually share the same area. Means your pollen from your cultivated rice can be pollinating your wild rice and is receiving introgression constantly. That's why you saw this heavy admixture. So with this admixed or non-pure population, how do you believe the conclusion coming out of it is solid? So we decided to explore better, and it's also reported. Wild rice is hybrid swarm with gene flow from domesticated rice. So we want to partition that introgression and just focus on the one that is actually the wild rice. So can we isolate the introgressions and then remove them from our analysis? So I use the hidden Markov model to identify ancestral regions from the two rice population using indica 3A, OSH, and temperature Japonica as the donor and OR2 as the receiver, and analyzing OR1 as the top track and OR3A as the bottom track, and see whether these regions derive from introgression or derive from the wild population. So the pink color is the wild population, and if I look at those shared between the two wild population, they are not a lot, right? Some of them dominating OR1, but some of them dominating OR3A, and they do not share a lot. For those shared, the genes tend to be about survival, like abiotic stress development, leaf development, and nutrient uptake, and even this lazy one is about architecture. So those remain as the wild region in both rice populations. They tend to be survival. So that means our analysis seems to finding something relevant. So other than that, I want to remove those. So I use this double pulse, double admixture model for OR1 because from the admixture, we know there are at least two donors. So I want to infer when and how large was the donation of genetic material to the background of wild rice, making our modern day OR1. So the number here was generation time with confidence interval of 95%, and with the proportion of genome donated to the modern OR1 genome. So you see, there was one pulse of around 12,000 generations ago, and then for the indica 3A was 4,000 generations ago, and each of them donate quite significant portion to the OR1 genome, making the nowadays observation. For the OR3A, it's only one donor, but I hypothesize it's a constant or multiple pulse of donations. So I use three pulse, a single addition model, and then infer the largest donation was actually 8,800 generations ago. That may answer your question. That they bounce back a little bit, and with the
Those are the posterior estimate. Those are the
posterior estimation. And then with the donation as early as 16,000 generations ago, and donate like almost 10%. And if we plot the generation time, and that visualize the two pulse donation from OSH, and then the major pulse donation from temperature connected to OR3A, and then the single large pulse from indica 3A.
This is great. You've answered a lot of my questions that I had before this with these slides. One residual question right now is, all of this analysis based on SNPs, or is it based on any larger variants?
Just SNPs, because we do not have
So
high confidence.
That's a risk for you, right? Because you may be missing a lot of important regions that are introgressed because even something as basic as inversion. Would you see an inversion?
We will see an inversion, but the mutation rate will be different because there's restricted recombination. Yeah, that's a caveat.
A bunch of retrotransposons here, you wouldn't see.
Yeah.
Okay.
Yeah. That's the title of what we can learn and cannot learn from sequencing data, right?
Okay.
I'm being cautious.
That part of the talk.
Yes. So how would the results look without the introgression regions, right? Now we identify them, so we remove them, and then I reconstruct the effective population size. And since they're all wild rice, I just convert them into years, 1 to 1. And you see this, still see the initial bottlenecks coming from OR3A, that's here around 10,000 years ago. But OR2 has an earlier, like 15,000 years ago as a bottleneck, and OR1 has a much later, which is like 5,000 years ago bottlenecks. And if we just use the SNPs coming from these ancestral regions and reconstruct the phylogeny, we have a clearer picture and seeing OR1 being a subgroup, a sister group of OSH. So they may be feral rice. And then OR2, OR3A, and OR3B being closer to the roots, and which is much better than what we just saw at the beginning. So, so that gives us more confidence on saying maybe it's only domesticated only once, but maybe with fertilization or maybe introduction from other wild rice, making the indica very diverse.
But this is before any evidence of agriculture. You're talking 10,000 years ago. There's no agriculture anywhere 10,000.
What define agriculture, right? So basically, it's starting from hunter-gatherer, right? You gather rice and then you spread it in your backyard. That's agriculture, right? So we don't know. And if you trace archaeological evidence,
Hunter-gatherers don't do that, though. Is there any evidence in India that any hunter-gatherers were reliant on rice 10,000 years ago? Or
yeah, that's the initial domestication of rice, and you always start with gathering from wild, and then maybe you watch them, and then they occasionally just like.
Well, and 15,000 years ago.
We have they are archaeological remnants, and they are collected in clay pots, and they are charred rice, and then you can date them, and then you can study their morphology, and you can define whether they look like a cultivated rice or white rice. And 10,000 years, they're already a cultivated form, but 15,000 years, they look like wild rice, but they're collected in a clay
So
pot.
that's way earlier than the standard Western European Turkish origin of agriculture or the Mesopotamian origin of agriculture.
Around the same time, 10,000 years. Yeah, wheat domestication, barley domestication around the same time, but maybe with some variation, like with some confidence time. So
I'll have to check.
yeah, please.
Rob,
Yeah.
we have the human selection information from amylase across Europe. It'd be interesting also to look at the one in East Asia, in the region where this cultivation was happening. But it looks like the peak of selection is around 9 to 10,000 years ago also.
So Eric, when do you see amylase blow up?
Yeah, it's a similar time frame.
Okay,
Mhm.
all right, I'll shut up.
No, this is a good discussion, and thank you for the questions. So yeah, the lesson learned here is isolation of cultivated rice introgressions supporting OR3A as a sister group of Asian rice, and then cultivated rice polluted the wild rice gene pool, but natural selection likely kept the wild genes that happen to survive in the wild. So the last question I want to explore here is, or I don't explore when, but where was the domestication of cultivated rice? So here is the geographic distribution of wild rice being collected. So they mainly just in the swamp or in the wild. But the question here is how were the wild rice distributed in their natural habitats? Remember we have several wild rice groups. So then I decided to study migration and introgression, like because rice do not walk themselves, the human carry them and then migrates, right? Here I use the stepping stone model. So basically, if you want to migrate from the square on the right to the square on the left, you have to walk several steps, and each step is your stepping stone. And the further away you migrate from your original site, the less similarity or the more dissimilarity of your genetic material will be. So this is the basis of the stepping stone model. And I used the estimated effective migration surface or EMS method published in 2016. Okay, so this is testing the model to see whether the distance between stepping stones is possibly created, possibly associated with this similarity between populations. And there's a general positive correlation. It's not perfect, it's not linear, but positive. So that gives us some confidence on using this stepping stone model to infer migration of rice. And this is how it looks like across the South China, Southeast Asia, and South Asia area. And And this is the South China Sea, right? You see there's no migration because there's no people walking in there. And this is the Himalayan mountains, and there's no migration because it's so difficult to climb up there. And you see this blockage of migration surface, and with the warmer color, less migration, and cooler color, more migration. And being the O3A mostly confined in South China, and we have two samples here, migrate through China-Myanmar border through here. So if they migrate or distribute to other region of here, then they through here. And OR1 mostly in India or in Nepal. And if you look at the genetic diversity, with higher color, cooler color means higher diversity. Yeah, there's also a low diversity zone confined by the geological barriers. And this is OR3A and OR1, OR2.
The previous, I didn't really quite understand it. So we have 5 groups or team, well, they're subspecies, it's called. And I don't quite understand. I see the pots, the points where they've been collected as a team.
Yeah.
I don't understand the migrate. I don't see arrows telling me how they migrate.
Yeah, there's no arrows. There's no arrows. Yeah, just so the model here is assuming the further away they're less similar, and if you migrate through these routes, you will have a gradient of your similarity. But if you don't see the gradient, that means you have less migration. So this is your migration circuit. Like this, there's a blockage here.
And red is absolute blockage, the Himalayas.
Yes.
Okay, so now if you go back, maybe I can understand that. So through the center of India, there's a blockage because it's a great big uplift of nothing but volcanic ash, essentially. And as you pointed out, Himalayas, well, that's actually below the Himalayas. So the red extends, doesn't barely extends into Nepal. No, if you look at Nepal, no, over there. Over above India. So then.
Center of the country is very dry, not conducive to rice. The place that you're seeing that's not very conducive to rice is very dry. And the blue part on top that's kind of tracks the Indus Valley and on the top.
Yeah.
And what is a little bit surprising is that the Gangetic Plain is not connected here. I mean, although North India does eat wheat more than rice, but I would have expected some movement there, but it is what it is.
Yeah, so if they consume less rice, it means that farmers less likely to carry them and share it with your neighbors. So they may result into a less migration. So this is just like.
This is just about migration. It's not where it is, like where it actually grows. It's just like what's the likelihood for a variety to move away from where it was originated. So it's not just a distribution.
It's kind of a heat map of mobility.
Yeah,
Yes.
you have mobility. There's no direction, but like you're more likely to mobile around this area, but not outside, right? Yeah. So if you look at the genetic diversity, they kind of agree. Like so you have more mobility in here, but not in the Himalayas, and not in the oceans. Okay. So then we want to find what's the possible initial domestication center, and based on the tree, we trace the rice samples that are closest to the cultivar tree at here. And with just a few samples, not a lot. And then we plot it on their actual collection. And this is all in China because it's OR3A. And Dongxiang is the wild rice variety that is collected the northeast so far. And this is how north they can be. But the cultivated rice they can go up here in the northeast China. And also Nanning has more samples distributed around this area, and there are archaeological remnants unearthed that is around 15,000 years ago in the clay pots that look like wild rice.
I'm familiar with that story, so that's
Yeah. So suggesting, so there was a debate whether it is domesticated in northern China or southern China, and if it's in southern China, whether it's China or India, right? But it tends to support domesticated in China and also southern China because we have more samples at this clade than just one sample in Dongxiang.
Okay.
So the lesson learned is OF3A is geographically isolated from other wild rice populations. And then the Guangxi region has more OF3A distribution or collection. So this is auto biased by the collection. And the Yunnan-Myanmar border are likely serve as the portal for rice migration out of China to South Asia. So the conclusion is that rice is likely initially domesticated as Japonica, likely, I don't have high confidence, with all the caveats of only SNPs and remapping, and more evidence support OF3A as the sister group of Asian rice. And then rice domestication is likely around 15,000 years ago to 13,000 years ago, with the correction of generation time. And if much uncertainty remains due to the heavy cultivar white rice introgressions and also use of short read and the sequencing data. So the future direction is to publish it as a final project of my thesis, make a conclusion of my PhD, and then use the rice pangenome assemblies, the actual genome, to study this question again to reduce uncertainties from short read mapping and the lack of knowledge from transposable elements. And then to use ancient DNA, if we can extract any DNA from the charred rice remnants to study the genetic composition of rice at different age of their domestication. And with that, I want to thank my PIs, starting from my undergrad, PhD, and my postdoc that all supporting the development of these projects, and with support from the Ecology, Evolutionary Biology and Behavior program and plant breeding genetics and biotechnology program in Michigan State, Department of Agriculture, and the Compute Center at Michigan State, and also the EXCEED now become assess of the extreme like large competition resource support and my current employer. With that, thank you. Question? Yes.
No, if someone else has a question, because I've been asking so much. The students first, of course. Perhaps I can get started and then we see. So two things. One is like about, I have two questions. One is about the time of the domestication. I just wonder if it could be inflated by something, like, you know, because it seems to me, I'm not aware of when domestication started in the in India or like in Asia in general. It's just because I don't know the history. But just like wonder if there's any kind of inflation. But you can think about it. It's more like a comment. Number two, if you ever thought about actually assessing selection and two types of selection, like you can think about a background selection on gene that have been introgressed from the wild rice, or just positive selection. Sorry, a background selection on the gene that remains from the wild rice, like the one that's being conserved, and positive selection for genes that contributed to, which we have seen already. And I don't know if this has already been done or you're thinking about it.
Yeah, it's done. It's just the next slide. I don't include it here, but I also scan for like sections and try to distinguish the type of sample, whether it's selection based on standing variation or selection based on the normal mutations. So I use a machine learning approach here because the other methods based on human data that assuming outbreeding. So colorate rice is inbreeding. So you have to consider that impact to their selection pattern and how long that can sustain. So yeah, I use this is simulation based. So I simulate hard strip and soft strip and neutral patterns, and then use a machine learning with the parameters from infer from rice. And then I use this classifier to identify soft strip and hard strip from the pattern. And we found very interesting things, and that is like the second half of the study. I decided not to include everything to just keep the story focused on.
Yeah. That'd be interesting to see, you know, once you have determined the order of the domestication, it'd be interesting to see which genes actually.
It will be still challenging because you don't have the clock on each SNP. You can infer, but that will be not very accurate. and lots of the SNPs are derived, into polygon. So the focus on the selection part was like what genes were selected in which population, and were there any focus on the domestication or improvement of different groups? Yeah, we found some interesting patterns. Yeah.
I know Cyrus is really itching to ask you some questions about transposons.
Actually, we already talked about.
Since you told us we were gonna have some transposon text. Go ahead and ask my question about transposons.
Actually, I don't have any question right now. So, yeah, I talked with Dr. Matthew a lot.
Yeah, so transposons likely ignored here because of the mapping of short reads. You don't have a high confidence on mapping short reads.
Everybody wants, don't feel bad about
Well, my lab
That's
tried
why
not
it's
to
so tight
ignore
about.
them. Yeah. So my lab tried not to ignore them right now, and but still, like using short read data, you can only study so much, right? Extract so much information from it. Yeah. So yeah, transposons are not considered.
Next frontier.
Yeah, that's the next, right, like use pan-genome assemblies. Right now we are already several hundred genomes of rice available.
I don't know if there's someone raising their hand from the.
Oh, that's just the.
Because I'm here, okay. So you're planning to do long read sequencing, right? Do you have
Yeah, those pan-genomes are all wrongly sequenced.
a long read? Oh, okay,
Yeah.
sorry, it was.
But my work right now is not focusing on the domestication question, but more on the transposal question. Like maybe a point would be like how transposons play a role in domestication. But that would be maybe some witch hunting process, like do you see any T's in the process of domestication? There is, right? Like in maize domestication, the flowering time, T's play a lot in affecting the flowering time genes regulation. And maybe we see something like that in rice.
Can you go back to your mixture analysis plot or map of chromosomes? One that actually showed the chromosomes. That's it. So they only have 12 chromosomes, is that true?
Yes.
Okay. Do they have, sorry for the naive question, do they have sex chromosomes?
No.
Okay.
Well, the gene, there's a gene correspond to isolation between the two subspecies in the certain chromosome. So if you consider that isolation is, but no.
So, yeah, this is where my question is. That slide, the one you just said. Okay, so who do you think is doing the selection on rice? Bugs or humans?
On cultivated rice, I think it's human.
Okay, so what there is human?
None of these looks like human. These are the
Why aren't they? You'd expect if anything is
yeah.
selecting rice
yeah.
and introgressing a particularly advantageous gene or haplotype, it's gonna be a human making that decision. Can I digest this rice? Does it taste good?
Mhm. So this looks like more about the fitness in the environment, like a
But there, I think Enza touched on this. There's positive selection, there's negative selection.
Mhm.
You're talking about negative selection here, right?
Yeah.
They need to be fit, they need to be able to reproduce.
Yeah.
But a human is doing more intense selection.
Yeah.
We selected dogs to be
Mhm.
docile relatively, to guard our homes.
Yeah.
And that they're a little bit different than wolves.
Mhm.
That was a positive selection
Yes.
on our part. And the
Mhm.
question is, given Eric's work on amylase,
Mhm.
we know that the humans were selected by their food source.
Mhm.
The food source was selected by humans.
Mhm.
So I guess the question is, there's a flip side to this whole slide, which is, what did humans do to rice genomes?
That's the second half of the story that has not is not presented here. But we found lots of selection on the nutrient uptake, like nitrogen, because rice is a fertilizer eater. So the entire nitrogen uptake, dissemination, and also transporting pathway, different part of them being selected in different population. We saw that.
That's a good thing, because humans will be adding nitrogen in
Yeah.
the form of dark soil, they call it.
Yeah,
Night
yeah.
soil.
Mhm.
Yeah.
Yeah, and even modern agriculture use chemical fertilizer. Yeah, and also the disease is also another pathway that look at. They usually act on, there's no systematic selection, but all on the sensing the first responders. The deeper layer seems there's no selection, which is also mostly on negative selection.
And rice paddies, I don't know if they have this in all parts of the world, but in Japan, they typically have monogamous species. So fish that are specific to rice paddies.
Yeah.
Is that true in China too, that there are
Yeah,
species
yeah,
of
yeah, yeah.
fish?
Yeah, right now we raise crawfish, not fish, crawfish, or yeah, in Louisiana, like lots of crawfish eating in the swamps.
But is it, well, but I mean specific to rice paddies, not just out there.
The fish, yeah, it's a species of carp.
It's adapted to Chinese rice paddies only.
I don't study fish, so maybe there's some adaptation, but usually people just use that species and then they tend to live pretty well in
And if they actually, if you start up a new rice paddy, would you say, I'm going to plant my rice, figure out how to irrigate it, and then I'm going to take some fish species.
There's no specific fish variety. It's just that carp species usually used in the paddy. Is it not
People put it in the rice paddy.
Yeah.
Intentionally.
intention? Yeah,
Okay, so
but not everybody. Not
That's
everybody.
another selection.
Yeah, that's another selection, but not everybody is doing that. So I think it's some practice, not all utilized.
Coevolution.
Yeah, yes. And including the soil microbe, right? Like in the paddy fields, you have submerged, low oxygen. It must be some special microbes. That's for indica, but for the japonica, they grow in dry lands. Like they just submerge for the seedling stage, and when they grow up, they dry up. So like that soil microbe is very different. And there's a researcher that I visited in Georgia. They study like microbes and very different.
Well, I look forward, when are you finishing your dissertation? Or
2018.
I'd love to read it, actually.
Yeah. Yeah. So this project keep haunting me, right? Like wake me up in the dreams, and like, okay, I still have it undone. Like, but I still see the value. Like we have some discussions, and so yeah, I want to finish it.
Sneak in some slides on human migration to neutralize my question, because I think of it as starting in Mesopotamia, kind of agriculture percolating out in both directions. But that may be totally wrong, because you're right, that southern part of China, I forgot completely that had a very early.
Yeah, there are several domestication centers right around 10,000 years of that time clock. Yeah.
So as you said, that all the ancient rice samples were found in like a very specific big pots and charred.
Yeah.
Why were they charred in those big pots? And
Oh,
did you see like that kind of like vessels?
Yeah. There are two reasons. One, they are too long, that they just naturally char. The second reason is they use rice stalks to make the house, because it's a filler. They use clay mixed with the rice stalks to make a wall. And then after make it, they burn, right, to solidify it and then char it. So there are two reasons, but the rice kernels are so small and they're very susceptible to all the environmental changes. And 10,000 years can happen a lot of things.
So just the rice was not like consumed, but like used to like.
Like they consume the rice and use the stock to make the house. Yeah, or burn, right? Yeah. And the remnant in the clay pot may be due to time, that or maybe they are used as like a berry serving, and then they also burn it. So, but most of the rice remnants are charred. Yeah, so that make it difficult to extract ancient DNA from it.
Probably because humans are, pillaging and burning down villages all the time, right? That's our most popular activity.
I don't know that. Yeah. Other questions?
Well, everybody have a good rice evening.
It was Indica, by the way. I think it was Indica.
Was there rice in there?
It was
Yes.
Indica. I'm sure it was Indica.
Yeah, it looks like long grain.
Like, yeah.
Yeah, yes. All right, so thank you so much.
Have you guys worked together before?
Sort of, have a project.
Thank you so much.
Did I understand you had already sequences